This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 144, recorded on July 28th, 2011. Hi, everybody. I'm Vincent Racaniello, and it's time for TWiV, the podcast all about viruses. Joining me today from North Central Florida is Rich Condit.
Hi there, Vincent. How are you?
I'm doing well. Very well, considering.
Good.
It's a nice day out. Probably nice down there as well.
Yeah, as a matter of fact, it's 92 and partly cloudy.
Nice. And you're going sailing tomorrow.
I'm going sailing tomorrow. I'm going on a big overnight job.
Nice.
We're going to be on the water for probably, 24 or 30 hours, something like that.
How many feet long is this boat?
30.
It's pretty good.
Yeah.
And you have a single mast, right?
Single mast.
Okay.
We're working up to a Bahamas cruise sometime.
Nice. I'll tell you, let's bring in from Western Massachusetts, Alan Dove.
Good to be here.
Welcome.
Yeah.
Missed you last week in Minneapolis.
Yeah.
We had a good time, right, Rich?
We had a great time. It was good.
Sounded
fun.
like
It
it.
was a fun meeting, yeah. Busy.
Meeting was great. Great science.
I can't remember ever being that busy at one of these meetings before. All the pox talks were spread all over the place. Plus there was TWiV and other things to do. I was working the whole time.
Great science. Amazing that we have such strong virology given the funding difficulties.
Yeah, I noticed that as well. It's all, people are doing the best they can, and they're doing a good job.
Maybe there's going to be a lag or something. I don't know.
We'll see.
But the TWiV was great also. We had two good co-hosts, and we had good science. The audience was great. They laughed. They cried. So, thanks everybody for doing that. And today we have a couple of good stories. Before we do, I want to mention the August schedule for TWiV, because it will be slightly different.
Which is not to be confused with the usual August schedule for TWiV.
That's right. Just makes a big difference where you put the accent.
Yes.
Doesn't it?
Don't want to put it on the wrong syllable.
Nope, we don't. We will not have TWiV next week. What's the date on that? Does anyone
5th of August, I think.
August 5th?
Oh, that's when we would record it.
5th,
right.
7th. So the 7th would normally be released. So no TWiV on the 7th. The following week, which is the 12th, right?
Right.
Right.
There will be a TWiV. And then the week after that, there will not be one. All right. So over the next 3 weeks, you'll have one TWiV. And for those of you that say we do it too frequently, this is your chance to catch up.
Everybody gets a little break.
For those of you who say we don't do it enough, I'm sorry. You have to go back and listen to an old one. But we need to take a little break. Okay? So That's that.
Okay.
Now, before we start our two stories, I wanted just to discuss briefly the idea of live tweeting or live blogging at science meetings. Now, live tweeting or blogging is when you have a laptop, and as the talk goes on, you post little updates. If it's Twitter, you do 140-character updates. If you're blogging, you could do a little more. Now with Google+, you could do a lot more. You could just summarize what's going on in the talks. This happens a lot at tech meetings. And I thought I would do it at ASV. They had good internet in the auditorium. I did it for the main talks in the morning. And I guess I did about 10 or 15 tweets per talk, but just summarizing the highlights. But after I did one session, someone came up to me and said, "Hey, I heard that you broadcasted my talk this morning." It's one of the speakers. "My student said you broadcasted the talk." And then later on, someone was saying, "Why are you doing this? Are you allowed? Did you get permission?" So I wrote a post on this over at my blog, virology.ws, and there's some pretty good commentary there. So for virology and other science maybe, but certainly for virology, this is pretty new. And I was wondering what you guys thought. Should we be doing this? Should we not? Does it violate something? Do we have to have permission?
Well, I posted a comment on your blog post that you probably saw. Right. This is something that has come up for me for years because I've done conference coverage as one of my regular things. And the live tweeting or live blogging makes it more immediate, but it doesn't really raise any fundamentally new issues. People at conferences like to talk about unpublished data because that's part of the point of a conference. If you're just going to get up there and review your papers, then why bother? But at the same time, you present unpublished data and then somebody reports on it. And if they report it in enough detail, it's possible that when you do submit the paper, the journal will bounce it back saying, oh, this has already been published.
Right.
And that's what's known as the Engelfinger rule, after the editor who came up with it quite a while ago.
Engelfinger, huh.
Yes.
Okay.
And it was originally...
That's a different compound than a zinc finger, right?
That's a different compound from a zinc finger, yes. Or any other kind of fingers you might have heard of. So the intent of it dates back to kind of the beginnings of science journalism in a sense. So there weren't always people writing news stories about the latest research. Mostly this stuff just went out in the journals, and the people who wanted to hear about it read about it there. But then as newspapers and magazines and everything started increasing their coverage of this sort of thing, there was a perception by the medical journals that they didn't want bits of information leaking out through the press that weren't part of the peer-reviewed study, and then the peer-reviewed study comes out and actually says something else or has a lot more nuance to it, and then you have to go and try and explain to the public, hey, those news stories that were on the front page a month ago were actually wrong because what the study actually shows is this. And we've dealt with some of this here on TWiV too, where some information leaks out and it's incomplete and you don't have all of the data, but people leap to conclusions, and then you have to kind of try to unteach all that information, and it's hard enough to teach science to the public in the first place. So it was a well-intentioned rule, but it raises this whole issue of how much can you report about a conference, and this is just always something that for science journalists it's been an ongoing problem. You want to report what happened at the conference, but at the same time, you don't want to tick off people who you'd like to use as sources for future stories.
Yeah.
So I, having come from a science background myself, I tend to err on the side of asking. And if I'm going to talk about somebody's work, I'll usually at least shoot them an email and say, hey, I'm writing this conference coverage. Was there anything in your talk that you really don't want out there?
Yeah.
But that's not, most meetings actually don't have very detailed policies on this. And I think you discovered at ASV, they didn't have any specific prohibition on it. So a lot of reporters will take that as, hey, it's a public meeting. Somebody says something at a public meeting. It's public information.
One of the things they got in the one sort of proviso on this that doesn't really specifically cover the tweets, that's in the ASV, the conference book.
Yeah.
Says information presented at ASV meeting or contained in the scientific program and abstract should not be cited as publication.
Right.
Material can be cited as personal communication only after the consent of the senior author has been obtained. Photographing, audio taping, or videotaping of presentations by audience members is disruptive and is not permitted. Okay, so basically you're not supposed to, in a scientific publication, cite something from the meeting, but that's not what you're doing.
Right.
And it also doesn't say that the photographing is prohibited for sort of security reasons. It's because it's disruptive.
Right.
And as one of the commenters on your blog said, what's this? This was a speaker who was in favor of it.
Right.
Said, we were told ahead of time that talks would be recorded and made available to the public in DVD form and had to sign permission agreements. So choice to present unpublished data was at our own risk. Now, I suppose, Alan, you raised the thing that if the whole talk is published, that's different because at least it's represented in its purest form. Okay,
right.
So there's no possibility that some little bit's going to get out and be misinterpreted.
Right.
On the other hand, people went to the meeting. They're going to talk to other people. They're going to, mess it up when they talk to people. it's going to get twisted. So in a way, I don't see the difference. It's just more immediate and maybe broader.
Yeah, that's a good point. There is actually there's a PLOS computational biology article on this. Live coverage of scientific conferences using web technologies. And what they say is it doesn't change qualitatively. It just speeds up the dissemination of information.
Right.
Right. And there are a lot of conferences now. Well, maybe not a lot, but there are some conferences that are doing live webcasts or that are doing delayed webcasts. And I do some work for the New York Academy of Sciences, doing meeting coverage for them. And in that case, whenever I'm working for a conference organizer, it's almost always the policy that my write-up gets checked by the speakers. Whatever I'm saying about their work, they can say, "Oh, don't mention this bit," because the conference organizers want to stay on good terms with the speakers. But the New York Academy of Sciences does a lot of stuff where they just put up a webcast. And they'll have the slides and audio of the speaker, and they'll often have a write-up written by somebody like me for people who don't have the time to watch all the talks. But if you prefer, you can go and you can watch all the talks. Mm-hmm. But at the same time, a lot of journals have backed off a little bit from the strict interpretation of the Engelfinger rule. So a lot of these talks that have already been webcast, people can then subsequently still publish, because the journals are acknowledging that this rule was never really a good solution, and it's become an even worse solution. And so there's an acknowledgement that, yeah, we do want the information to be accessible to the public, and that is the way things are done here in the 21st century. But it's got to be, there are still some limits on how it's done, and everybody's trying to figure out how that's supposed to work.
I mean, it's weird because, can you tell somebody, and we're talking about live blogging or live tweeting here, okay? And so maybe if you decide that that's not good, you could tell people you can't do that. Are you going to tell people that they can't, a day after the meeting is over, go tweet some result? Right? I mean, where are you going to draw the line?
Yeah, or maybe not even a day after the meeting is over, just an hour after the talk is over.
Right. So I think this is going to happen, right?
Yeah.
And so it just, people are going to have to learn how to deal with it.
Yeah, the only exception would be something like a Gordon conference, where there is and always has been just a blanket prohibition on that sort of thing, where they say, okay, it's a Gordon conference. You come here, you talk about your science, but the rule is you don't talk about the conference.
Right.
Yeah, I think it's probably a good thing, and that's why I did it. And a lot of people said they enjoyed it because they couldn't come to the meeting. Right. And if you look at the comments on the blog post, a lot of people are in favor of it.
Yeah, most people are. It's mostly positive.
Open science.
The other thing that came out of the blog post that really shocked me is the notion that a number of libraries no longer have print of journals.
Oh yeah. So in our library, Alan probably remembers all the journals.
Yeah.
Those are gone.
Yeah.
They're gone.
That is an absolute tragedy.
They replaced them with computer stalls and study areas. Yeah.
Oh, that is so not equivalent.
I agree.
Wow.
I mean, I think they ship the older books somewhere else, but they're not there any longer. You could go down 3 or 4 levels and get into these old journals from the 50s, 40s, 30s. And
working on polio, that was great. You could go down there and you could find original papers by Sabin and Salk and look at their materials and methods, and it wasn't some horrible scan of a tertiary photocopy of something where you're squinting at bands. It was the actual printed journal. You could look at the thing.
Yeah, that TWiV article that I sent to you guys, I got from the top floor of our library. Lancet, 1915. It's terrific. Wow.
Yeah, that's another issue altogether, but I think open science is the way to go here. Someone here says, you have no idea how exciting it was for me to receive your tweets throughout the meeting, which I could not attend.
Yeah, a lot of people are very positive.
I mean, I think they want to hear what's going on. They don't want to steal your data.
Yeah, and there's no way that you're going to disclose enough data in a tweet.
I think not.
A blog post, maybe. But again, it gets back to this point that if people are presenting this in a meeting, they want people to know about it. If you really want to keep your results secret, then keep your mouth shut.
Well, look, someone said to me, they presented a protein that they had pulled out of a screen that they haven't mentioned before, and they said now it's out there and everyone else can start working on it. And I don't know. I think eventually you want to disseminate the information. Are you worried about someone getting a jump? My philosophy in science has always been, if I can't finish something that I started, well, I wasn't good enough to do it. And that's fine with me. I know everyone doesn't feel that way, but I think if you talk about something at a meeting, you're going to have to assume if there are 1,000 people at the meeting and they go talk to 10 people each, it's pretty good dissemination.
Yeah, it seems to me that if you're worried about something getting out, just don't talk about it. The people who are most likely to want to scoop you or whatever or work on the same thing are right there at the meeting.
Right.
And I mean, this person who was worried about it probably forgot that the talks were recorded and you can buy the DVD.
Right.
You can go to the website and buy it. And then I'm sure some people are going to rip them and put them up somewhere, which is, I'm not condoning it, but there are lots of ways that this can happen. And personally, I think going forward, this is going to happen more and more.
Yeah.
So I'm in favor of it. I just was a little worried that there was this reaction to it. But when you poll the broader audience, you see that I think going forward, as Marco said, when the PIs who are used, when the people who are the Facebook, Twitter generation move up into the ranks of PIs, it'll be a lot more easy to do this.
Yeah.
Right? And as we old guys, he said, you are among the first to do this stuff, but there will be countless more. Yeah. In fact, I must say the people who complained to me were slightly older, let's say. I won't call them old, but they are slightly older. So anyway, I thought that was worth touching on.
Yeah.
Yeah.
So we'll put a link to that blog post and you can make some comments there. Check them out yourself. All right, our first story is actually not based on any journal article, but a variety of stories in the press, which is fine. We've got a few readers who have sent us articles. And this concerns Hendra virus. There's currently another outbreak in Australia. And we haven't covered Hendra since way back when, I think. We may have mentioned it here and there. We talked about it way at the beginning of TWiV. And in fact, one of the original veterinarians who worked on it in the first outbreak in Australia emailed us a long time ago and said he would love to come on the show and talk about it.
Yeah.
But Hendra virus, of course, is a newly emerged virus that appeared in Australia, and it is a virus in the paramyxovirus family. So it's an enveloped virus with an RNA genome, negative stranded, and related to, for example, measles and parainfluenza viruses. And it was first identified in 1994. There was an outbreak among racehorses in Australia. So this was respiratory disease. And the handlers of the horses, of course, the handlers get close and they get their secretions. 2 of them were infected and 1 died. And the horses can die as well. And if you're a racehorse, even if you're sick for a while, you're losing money.
Right.
So this is a big deal. So that was 94, and this virus was brand new. It was isolated from the horses, from the person who died. And it was called Hendra, so that's where the first outbreak was. And there have been 10 outbreaks since then. I guess the current one would be the 11th.
Right, but it looks like, I mean, this one, in one of these articles, it looks like the current one is a lot bigger than in the past. It says this year's total matches the total cases that Australia had seen prior to this year. Until this year, there were 14 cases over 17 years, and in the last 6 weeks, there have been 14 more nationally. Now, I suppose that could be a diagnosis thing.
Okay,
right.
But at face value, relative to what's happened in the past, this looks like a big outbreak.
So the infection goes from a fruit bat to the horse. The virus goes from a fruit bat to the horse. So fruit bats, those big bats, huge wingspan,
Flying foxes.
Flying foxes, 3 or 4 foot wingspans.
6 feet,
6
it says
feet.
in Wiki. Up to 6 feet. That's a big animal.
Yeah.
Yeah.
So they have this virus and they're fine. They don't get sick. It's one of the things they live with. And they can transmit it to horses, and who knows how, but probably the bats go in at night and eat some food around where the horses are, or defecate or urinate or do whatever, somehow contaminate where the horses are, and the horses pick up the virus.
Presumably by eating or inhaling contaminated material.
Right.
Right.
So, this happens when you build horse farms close to the forest, right? The forest is where the bats live. You impinge on the bats' habitat. So this didn't happen before because we didn't do that. And as we move into their ecological area, and maybe that's why there are more now. Maybe some of these are new farms. Who knows? But might be, as you said, Rich, it could be detection as well.
Mm-hmm.
And
or there could be a change in ecological conditions too. Maybe a drought or an unusually wet year or something like that that's changed the balance and caused the bats and the horses to be in closer proximity.
Right.
Yeah. Well, they noticed that they note in one of these articles that the transmission rate seems to go up when the bats are underfed. When they're having problems finding food. I don't know quite the connection, but there's an epidemiological correlation between when the food is scarce for the bats and transmission to the horses.
Okay, so maybe the bats are coming closer to human habitation to find more food.
Something like that. At least it supports your idea, Alan, that a change in ecological conditions can influence the transmission.
Right. But it still comes back to Vincent's point as well, that if you weren't building the horse farms right up against the bats' normal habitat, you wouldn't have this problem.
Mm-hmm.
We should also point out that human-to-human transmission has not been seen.
Right.
So you get it from the horse, and since you probably get a good inoculum from the horse, you get sick and it can replicate in you, but it's not enough to transmit to other people.
And it sounds like the transmission from the horses to humans is not real efficient. Okay,
no.
They got people getting infected who are performing autopsies on sick horses and that kind of stuff.
Right,
well,
and trainers who are handling horses all day.
Exactly.
Right.
Well, even the horse infections are pretty rare. I mean, this is it's quite rare, and I think
because they find it in a lot of bats, right?
Yeah. In many, many bats. And
we're talking here about an epidemic, and it's 14 cases of horses.
Now, is it the case that this has only been seen in Australia?
I believe so.
I think so. Now, of course, there's Nipah virus in Malaysia,
right?
Which is related, but I'm not clear on how closely related.
Yeah, that is a different virus, but in the paramyxo family. Same idea, going from bats to pigs that time.
Right.
There's that's spread a bit more than than Hendra.
And is that one that has infected humans as well? Is that correct?
Yeah, it has. And there's some evidence for human transmission, but it's not established yet. But I think its scope is greater than that of Hendra, as far as geography. Although Australia is pretty big.
Yeah.
There's a really nice map of the current epidemic that I think I put the link in the in the show notes. Have you got that map?
Yeah, let's see. Yeah, it's going from Cairns at the north all the way down to Port Macquarie.
Yeah, I had to even learn some geography here because that's Queensland in the northeast sector and New South Wales in the southeast sector. Right. Most of them are in Queensland. They're all along the coast. Of course, I guess most of populated Australia is on the coast.
It's along the coast, yeah.
Now, one of the things that has been going on here is that some people say that, Peter sent us the following. Hendra, all the dummies come out of the woodwork. Kill all the bats, cut down all the trees.
Right.
Yeah, so Peter is down there in Australia. He sent us a link to an article. And they say, let's just kill all the bats and cut down all the trees around these horse farms.
Right. Some politician said that.
Yeah, because of course that never works.
No.
Although in the case of chickens and highly pathogenic influenza, killing all the chickens on a particular farm will stop the outbreak because you have
well,
no
that's
more
the difference
chickens.
it's the difference between domestic and wild animals.
Yeah.
If you try to eradicate domestic animals, that's actually quite easy. You know where they are; they're all contained. You wipe them out, and they're gone.
Yeah.
Trying to eradicate a wild animal, and to hear Australians talk about that in particular was just the peak of irony.
This has also been done in Europe for outbreaks of foot and mouth disease virus. They culled the cow herds. There's also BSE. They were doing culling of cow herds. But yeah, so when you have domestic animals, it works. But a wild animal, you can never wipe them all out.
Yeah, I mean, why don't they just wipe out all the rabbits?
Yeah, it's awfully presumptuous to think so. So Peter, who down in Australia, sent us a link to that. I have another email from Matt, who said, I've been following the latest outbreaks of Hendra in Australia. It has killed several horses, actually more than several now, without any positive results in humans so far. It appears the virus may be able to infect dogs, or at least can cause an immune response in this species. Not sure if this is a case of if we look hard enough we will find it, or the detection of Hendra moving into a new species for the first time.
Yeah, this is actually interesting. This is, in fact, that's what stimulated the stupidity we were just talking about with the kill all the bats idea. Was apparently a dog on one of these farms that had several horses die. The dog tested positive for antibodies against Hendra virus.
Right.
And that's what stimulated this discussion of, well, let's take extreme measures.
Of course, all the headlines say deadly Australian horse virus found in
dog, but it wasn't.
Right.
What was it?
They
antibodies.
Antibodies. They tested for virus, and one of the stories actually points out they tested for virus and the culture tests were negative.
Right.
But the antibody test was positive.
So we don't know how they look for antibody, but I would guess that they maybe have
actually they got an ELISA.
Yeah. And they used viral protein. They stick it on plastic, right?
Right.
Then you pass serum over it, and if there are antibodies, they stick.
Right.
And you can imagine that maybe other antibodies to other related viruses might react, right?
Cross-reactive antibodies, those are certainly well known.
So it doesn't have to be Hendra. It could be a related virus that we've never known that may have infected this dog, or maybe just cross-reactive antibodies.
Yeah, and very importantly, I don't think there's any denominator on this. They haven't screened all the dogs in Australia to see how common it is to have apparent antibody responses against Hendra virus.
Right.
It's possible that there's some dog virus that generates cross-reactive antibodies against Hendra, and that it might actually be common, and they just happened to test this dog because it was at this farm, and it came up positive.
I mean, the dog may not have been infected. Maybe he got a bolus of virus, and it didn't replicate, but made an antibody response as well.
That's also possible.
So they want to kill this dog, or maybe they already have, but I don't think that's indicated.
No.
Well, I'm a little on the fence about that. I see, on one hand, I having just talked about how weak the evidence is that the dog actually had this virus. It's just an antibody test. You even did the culture test and it came back negative. My suggestion would be quarantine the dog, test it again.
Sure.
And keep up, do more sensitive tests if you can, and really probe for the virus. And if it continues to test negative, maybe let it go. On the other hand, I can kind of sympathize with the public health authorities who, if you think about it, what if this dog really does have the virus and that it's just a silent infection, and now you've got it in an intermediate host. It's no longer in the flying fox. It's not in a horse. It's in the dog, which can survive with it. And if this becomes the nucleus of some future problem, then whose fault is that going to be?
That's nobody's fault. Viruses are a natural thing.
Right, but the public health authorities who made the decision to let the dog live are going to lose their jobs over it.
He's in quarantine.
I would say probably quarantine for a period of months, which they've already got the dog quarantined on the farm.
I think they should, as you say, they should check other dogs on horse farms as well.
Yeah, I would go around testing dogs.
Or other whatever animals. I mean, there are probably a lot of cats there. There are typically cats in horse barns because there are mice.
Yeah.
and check them all. But
So,
yes.
So this is a classic example that's, I think, worth pointing out of a zoonosis, okay? And we tend to think of zoonoses as animals infecting us because most people don't think of people as animals, okay? But a zoonosis can be an animal infecting another animal too, okay?
Mm-hmm.
So we have a virus that is in equilibrium with the bats, apparently. It infects the bats are happy, the virus is happy. And on occasion, if a sufficient dose gets into some other species, in this case, primarily horses and occasionally humans, it's kind of an out of balance infection and can cause really serious disease and maybe high mortality in that other species, but is unlikely to be transmitted very efficiently. Okay, so the horses don't seem to pass it to the horses. They can, on close contact, pass it to the people. The people haven't passed it to other people, so transmission is actually limited. This is classic. We see this all the time. I mean, what am I thinking of? The Four Corners Sin Nombre virus,
right?
And Ebola virus, and a bunch
avian influenza,
right? Same sort of thing.
So what do we do about this? You can't cull. It's not going to do any good. The bats are going to continue to fly into the horse farms. What should we do?
Well, I think the vaccine for the horses makes sense. Okay?
Yes.
Because the horses seem to be like the primary, secondary reservoir. I mean, if you could immunize the horses against this so that they didn't get sick or get high titers of virus, you might be able to stop it right there. You can't there's no sense trying to get rid of the bats. I mean, that's not ethical anyway. And it's not recommended by any of the public health people.
And it's probably not feasible.
Right. And the farmers, there's actually, in one of these articles I dug up, there's a lot of recommendations for how the farmers can protect their horses. You know, you put covers over the food troughs and that kind of stuff. You don't park all the food right out under the fruit trees and that kind of stuff. So you can take some simple precautions to limit the horse's exposure. But I think a vaccine for the horses, which is apparently in the pipeline, is ultimately will stop the thing, take care of it.
Yeah. Frequently with infections that are coming from wild animals into farm animals, the first line answer is better animal husbandry practices, and just prevent that contact from occurring. And that can be extremely effective until you get a vaccine. And in fact, one of the papers I think you linked to, Rich, was a very promising lead on a vaccine.
Mm-hmm.
Yeah, this is from a group in Australia.
Appropriately.
CSIRO Livestock Industries. A recombinant Hendra virus glycoprotein-based subunit vaccine protects ferrets from lethal Hendra virus challenge.
Right.
So apparently you can infect ferrets with the virus and it kills them. It replicates in their lungs, so it kind of
gives a disease very much like the disease you see in horses, apparently.
And now this is a dangerous virus to work with, so you don't want to grow it up and inactivate it to make a vaccine. So you just produce one of the proteins, and this is the glycoprotein on the surface that is probably immunogenic and which would be protective. And they immunize ferrets with it, and it's protective. So one can imagine this going into horses eventually.
Yeah, not only is it protective, but at the 2 higher doses that they used, they weren't even able to find the virus from nasal washes, which suggests it probably would prevent them from carrying it.
Right. And of course, the bar to licensing vaccines for horses is much lower than humans, right, Alan?
Right. I wouldn't think this would be a problem.
Because there already are other types of horse virus vaccines out there,
right,
which we've talked about. So I would guess that's the way to go, and I'm sure the horse farmers would like to do this. And until then, those precautions that you mentioned are worthwhile. Okay, anything else? Did we miss anything?
No, I think that's about it.
We have a lot of stories. We'll put all links to these in the show notes. You can check
this
them all out.
This one article that I found that sort of is a nice review from New South Wales does emphasize under prevention of infection. They got a couple of nice paragraphs here about all of the husbandry things that can be done. And they say, Flying foxes are a protected species, so culling is inappropriate and is unlikely to be a feasible or effective prevention strategy. Habitat loss and alteration, roost disturbance, urbanization, and being hunted stress flying fox colonies, which may magnify the problem. So people want to cut down all the fruit trees and that kind of stuff. And these guys say, nah, forget it. That stuff's not going to work.
So that's one lesson here, that culling, not a good idea. The other is, just because a dog has antibodies does not mean he or she has the virus.
Hendra virus investigations can generate intense media interest and may evoke fear amongst the public. Open and transparent communication is therefore important. An experienced spokesman should be nominated and all inquiries referred to them. Blah, blah, blah. Community meetings with public health professionals and animal health specialists may be necessary to inform the public on risks and outbreak management. So that's as important as anything. Don't flip out, dudes.
Hello?
You left us.
Yeah, I don't know what happened there.
It's all right. You're back. Are you there, Rich?
I'm here.
Okay.
All right.
All right.
Let's move to our next story. This one I've had for a few months, and the title is Engineering HIV-resistant Human CD4 positive T cells with CXCR4 specific zinc finger nucleases. That's a mouthful.
Yeah,
I'll say.
A lot of authors, too. Craig Weiland is the first author. The last author is Robert Domes, and many, many in between.
It's a lot of work.
Yeah.
This is an amazing paper.
This is an amazing paper.
Zinc finger nucleases. I got to tell you why I picked this paper. It's really cool science. But I got to diverge a little bit here.
I'm glad to hear this because I was wondering how you came up with this.
So my family loves this TV show called NCIS. Okay? They're always watching it. And I walk by, I see it, and it's actually not bad. I don't watch TV, but I will sit down with them and watch this. And there's a character on the show. Her name is Abby. She's a forensic scientist, and they're always in her lab. And she's got fridges full of Fisher bottles of chemicals and Eppendorf centrifuges. It's just like our lab. Okay, it's pretty cool. And there was one episode where she's talking with some agent from another place, and so she says, "I'm not familiar with Dr. Heller's work," and the agent says, "No, but he is with yours. He was quite impressed with your paper on zinc finger nucleases in DNA binding." And I'm listening. I'm sitting there with my family, and I listened to this. And the way he said it, you can tell he had no clue what he was talking about.
Right.
Yeah.
And I said, he doesn't know what he's talking about. And my family said, shut up. I thought it was so cool. I don't know where they got this or if they have a science advisor on the program.
Random PubMed search.
So I said, we have to do a paper on zinc finger nucleases. And here it is.
Yes.
This is very cool. Hey, Rich, do you like zinc finger nucleases?
Well, I really didn't care much before you pointed this paper out to me. I learned a lot from TWiV, and now I'm all over it. These are great.
Do you want to explain what a zinc finger...
Thank you for asking, because I really do.
Good.
Okay?
I got the sense.
This is incredible. So, zinc fingers... actually, there's a good wiki entry on zinc fingers. They describe a lot of protein structural motifs that are coordinated to some extent by binding zinc ions, okay? And they do lots of different things, interact with stuff. A subset of these are DNA binding motifs that consist predominantly of an alpha helix. So for the uninitiated out there, just think of a nice little sort of tube structure, a protein tube structure. And the interaction of these things with DNA is very well understood. So well understood, and I didn't realize this, that you can actually design in the laboratory a zinc finger DNA binding motif that will specifically bind any 3 amino acids in a DNA helix that you want. Okay?
3 nucleic acids.
Sequence of 3 nucleotides.
Right.
So I don't know what I said, but sequence of 3 nucleotides in a DNA molecule. So, GAT, TCG, whatever. You can design one of these helices so it'll bind that. Then, if you want to, you can actually connect them together in a single polypeptide. So this is entirely synthetic. Okay, you can string them together in a single polypeptide and put together an artificial series of zinc finger motifs that will bind a string of nucleotides in a DNA. So say you've got a sequence of 12 nucleotides that you would like to be recognized for some reason. You can string together 4 of these specifically designed alpha helices to specifically recognize that. Fine. So now we got a protein motif that can recognize a specific DNA sequence. Now you hook onto that a domain from a restriction endonuclease. Restriction endonucleases are site-specific DNA cleavage enzymes that come from bacteria. They are bacteria's immune system, okay? So that bacteria, when they're invaded by some phage or some foreign nucleic acid, can digest these things. So they have specific sequences because each bacterium contains a different one to retain its identity. One of these is called FOK1, F-O-K-1, which I gather is relatively nonspecific in its recognition. And you can, and it works as a dimer. So you take one of these and you stick it onto the end of one of these zinc fingers, the artificial ones, and you create another zinc finger that, say, recognizes another sequence in a, another sequence that flanks the sequence that you're interested in. So now you have, in the paper that's cited here, a 24 nucleotide sequence. So on the left-hand side of your iPod, you have a sequence of 12 nucleotides with a zinc finger binding to it, and then a FOK1 subunit and a sort of some DNA sequence in there, and then another 12 nucleotides on the right-hand side bound by a zinc finger thing in the opposite orientation, so that the second FOK1 subunit is dimerized with the first one, and they can cleave in between that sequence. So you design this thing totally artificial, and now it can fish out a 24 nucleotide sequence in whatever cell it's expressed in, which is likely to be unique in something as complex as the human genome, and cleave in between them. So you can design this enzyme to be expressed in a cell, find a completely unique sequence, and cut it. My God.
Right. So the enzyme FOK1 has a recognition sequence that is specific, but then it cleaves 9 to 13 nucleotides downstream of that.
Okay.
A lot of enzymes, restriction enzymes, as you know, are like that.
So they must have just the nuclease domain from that, not the recognition domain. I'm assuming there's 2 domains of the FOK1.
Yeah.
So right, because you're providing the recognition with the zinc fingers.
Yeah.
So exactly. A zinc finger nuclease is an entirely artificial construct.
It's brilliant.
It's zinc fingers hooked to a nuclease domain, okay, designed so that it can fish out a completely unique sequence in a complex genome and cleave it. And then, of course, what happens? Well, of course, what happens is in the cell, when that's cleaved, the cells freak out and try and repair it. And since it's, I don't know, it's not blunt, is it? It's
staggered. Yeah.
It's a staggered cut, but the cells can't put that together right. So they do a thing called non-homologous end joining,
right.
Where they try and at least stick the DNA back together, all right? But they, more often than not, screw it up. And you wind up with sometimes insertions, but more often deletions of a few bases. And the effect is to inactivate that gene. Okay.
Right.
Yep.
Right. If you have done this in a protein coding sequence of the gene.
Right. You're likely to get a frame shift. Even if you don't, you have several amino acids maybe that are deleted, right. And most likely you're gonna mess it up. So if you design one of these things and express it in a cell, it'll fish out a gene and genetically inactivate it permanently. Done.
Right.
What's cool is that you can put this nuclease in the cell, it'll do its thing, and then the nuclease can go away, but the genetic alteration is permanent.
Right. In the lexicon of molecular biology, hit and run.
Yeah, you don't need a retrovirus to integrate and stay there forever. So
do the job, go away, it's done for good.
Now, Alan, you can have a field day with zinc finger and FOC1.
Yes.
Okay.
I am exercising extraordinary restraint here.
I'm just pointing that out.
Yes.
What an amazing combination. Okay. Now, this paper uses it to try and make cells resistant to HIV. And we all know of this German individual who received a transplant of cells, bone marrow cells, from a donor who was deleted in one of the co-receptors, one of the genes that encodes the co-receptor for HIV. So HIV needs to bind CD4 as a receptor and also one of two co-receptors, either CCR5 or CXCR4. So that German patient received cells from a CCR5 mutant, and now he is free of virus because all his T cells now aren't able to bind virus. So that's sort of a proof of principle that this idea works. That you could, if you could interfere with these receptors or co-receptors, it might be a good way to resolve infection.
Right.
And now the problem is there are viruses that use or that bind to CCR5 or CXCR4 or both. So you really should knock out both of them if you want to do this properly.
And also you'd like to knock these genes out in ideally the patient's own cells, or because you can't just go around finding these elite controllers and using them as donors to repeat this experiment.
Yeah, good point. So that German fellow received a donor from someone else, but he was compatible with him.
Right.
But that's very hard to get.
So what you want to do is take a person's own cells and knock these genes out and put them back in the person.
Right.
It's called an autologous transplant, right?
Right.
Yep.
Take out your bone marrow, you culture it, you knock out these genes, put them back in. So I didn't realize there was already a clinical trial for a zinc finger nuclease that knocks out CCR5.
I did not realize that either.
Yeah. Now, but this paper, they're looking at CXCR4 because you have to do both, really. But going through it a little bit here is illustrative because this is amazing stuff. So they design zinc finger nuclease as much as Rich has said against CXCR4. There's a wonderful figure in there, figure 1. I guess they designed a one that recognizes 12 nucleotides, right, Rich?
It's total of 24, 12 on each side of the breakpoint.
And so they make a DNA sequence encoding this zinc finger nuclease.
This is open access, right?
This is open access, absolutely.
So I think that figure 1, at least the top part of it that shows the zinc finger nuclease binding to the DNA ought to be maybe the graphic for the episode.
Can do that,
right.
In case people didn't get everything out of my description there.
And by the way, now that you mentioned that, very soon, I hope, perhaps within the next few months, I would like to live stream the audio for TWiVs as we are doing it and allow people to ask questions as we're doing this.
Wow.
So they could look along on an open access journal article with us and say,
"What?
I don't get that. We could make a course out of TWiV." So you take the DNA, they put it in an adenovirus vector and then infect cells. And Rich, I'm sure you noted the multiplicities of infection.
Yes.
That they
huge.
Used. So they infected different multiplicities in cultured human CD4 T cells and say how much adenovirus do we need to get a certain percentage of breakage of the gene or disruption. Really huge. They decided to use 600 particles or PFU per cell.
Yeah, I was wondering if it was particles or PFU. Is that actually... Because that makes a difference.
They could do PFU because you can titrate these defective adenoviruses on complementing cell lines. I don't see how they do it.
You go ahead. I'll look back here and see if I can find it.
So they show that if you infect CD4 cells with these viruses, that in fact CXCR4 is disrupted. They measure that in a variety of ways. They either sequenced the gene directly or used what they call their surveyor nuclease assay, which I bet Rich would love to explain.
Yeah.
Just a quick one about the multiplicity. All they say is multiplicity of infection of 600. I would assume that's PFU.
Yeah,
right.
It could be either one.
For the listeners, that makes a difference because the particle that lots of virus preparations have a lot of particles in them that are not, by some assays, whatever assay you're using, biologically active. And it's not untypical at all to have particle infectivity ratios of 10 to 1 or 100 to 1. So if it's 600 particles per cell and the particle infectivity ratio is 100, then that's only 6 infectious units per cell. So that makes a big difference.
Right. I strongly suspect it's PFU because counting particles is difficult.
Right. Yeah, good point. Well, unless they're purified viruses, you just if you've purified the viruses, there'll be some sort of light scattering measurement. Just take an OD, and and that'll give you some concentration that somebody before has converted into particles. So, and there's nothing in there they don't talk specifically in the article about the preparation of the virus, so I don't really know for sure. Anyway, they dump a lot of virus on these things.
Before you explain this surveyor, so they get at an MOI of 600, 34% of the cells have disrupted CXCR4 genes. Now you may say, well, that's not 100, right? That's no good. But when the others that are not disrupted are infected, they get killed, and the ones that are left are the resistant ones, and they will grow out. So it doesn't have to be 100% effective. That's the cool thing about this approach, I think, or one of the many cool things. All right, what is a surveyor?
So the surveyor nuclease assay, this is a commercial assay sold by, I don't know if it's sold by other people as well, but there's a company called Transgenomic that sells this. And I was not familiar with this before. I guess I really am an old guy. But
is that the definition of an old guy? You don't know new stuff.
So the idea is you got these cells, you've made you think you've made a mutant. You got it. And it may be just a single base change or a couple of bases in one gene out of a whole genome. How are you going to find out whether you did that or not? So what you do is you make the DNA and you do a PCR reaction such that you amplify a piece of DNA surrounding where you think you made the mutation. Okay? And you do that both from the candidate mutant and from wild type. So you got 2 PCR fragments, same size, theoretically one mutant, one wild type. You denature those and then reanneal them, meaning you denature means the strands separate, reanneal means the strands can rehybridize. And in some cases, the wild type rehybridizes with the wild type, the mutant rehybridizes with the mutant, but about, I don't know, half the time or whatever, you'll get a wild type strand hybridizing to a mutant strand. And if the mutant has even a single base mismatch, that creates an aberration in the hybrid. And there is a nuclease called surveyor nuclease that is a mismatch-specific endonuclease derived from celery. All right?
Glad somebody looked.
Yeah, glad somebody looked. There's something that the NIH would have a little trouble funding. Celery? Okay, but this is cool. And this enzyme will recognize as little as a single base mismatch or more and cleave the DNA. So then all you got to do is run this stuff on a gel, and there's other assays you can do as well, but you can just run it on a gel and see if there's a certain fraction of the reannealed pieces of DNA that have been cleaved. Bingo.
Yeah, it's great. Another good reason to do good science.
Yeah, just do good science.
Yep.
Okay, so they.
And by the way, there's a good animation of this whole process on the company website.
Yes, very
good.
Nice. Yes, very nice.
Did you put that link in?
I think I stuck that link in already.
Okay.
Yeah.
Very good. So there are a number of different kinds of mutations that can occur. after this double-strand break, and they do some work to characterize those, which we don't need to go into. One of the interesting things they do is they want to know the specificity of these zinc finger nucleases. Besides cutting where you want them to cut at CXCR4 gene, do they cut anywhere else?
And that's important, because if they're cutting off-site, you could theoretically create unwanted mutations in your patient.
Right.
That's
right.
Side effects or whatever. I think this is a very interesting sentence. Potential off-target modification comprises the predominant safety concern with zinc finger nucleases. Although ultra-deep full genome sequencing could best identify off-target effects, it is impractical and cost-prohibitive with current technology. So in other words, you could sequence the whole genome.
Right.
And eventually we'll do that for $1,000.
Eventually you do that.
Right?
Yeah, sure. You'll have something looks like a PCR machine on your desktop. You just pipette some cells in there, and it'll tell you the genome out the other end.
Yep.
But now they couldn't do that. So they used a procedure which I think we spoke with Lieberman. Was that her name?
I forget what.
Judy
yeah,
Lieberman.
I think so. Yes, CELEX.
CELEX.
Right.
Where you identify nucleic acids that bind a protein, essentially. So they take their zinc finger nuclease and they pass it through a collection of sequences and see what binds to it.
Right.
And these are random sequences, which presumably are everything you might see in the genome. And you see what binds and you sequence it. And they found 2 sites in the genome. One is an extragenic region on chromosome 12, which means it's not a gene, but who knows whether it's important or not. And the other one is a metalloprotease of unknown function. So there are 2 off-site hits with this zinc finger nuclease. And so they don't really get back to this and say whether this might be an issue. Or who knows if you could have side effects or long-term issues after cutting that DNA, right?
Right.
But anyway, that's an interesting approach.
There's also the issue that's CCR5 and CXCR4 are doing something in normal biology, right?
Right.
Yeah, they do address that. It's very interesting. We can actually mention that right now. They say they're not really that important. Let's see where they said that.
As far as we know, yeah.
Okay, CXCR4 plays a critical role in normal B-cell cardiovascular and cerebellar development, though T lymphocytes appear to develop normally in null mice. So they say it may be tolerable.
Yeah.
So the thing here is that it looks as my sense was, and they have a wrap on this in the discussion, that CXCR4 may be necessary during some of the earlier stages of lymphocyte development and stuff, and not so critical later on. And so it may be, this influences whether you're going to try and engineer stem cells or some later non-committed cell or something like that, okay? You may not be able to knock CCR4 out of hematopoietic stem cells and get this to work, because you may screw up hematopoiesis. Whereas CCR5, not a problem.
But what they plan to do is take out your T cells, modify them, and then put them back into you.
Yeah, but they'd have to do it in a fashion where, you can get some permanent amplification of that. So if it's not a stem cell, I don't know enough about hematopoiesis to say what you could do.
Yeah, if you just take out CD4 positive T cells and then put them back into the patient, then after a period of time, those are going to die out and then back where you started.
Right. You need some sort of progenitor that's going to be able to regenerate itself, but doesn't really need CXCR4. And I don't know how big a problem that is.
Well, if you're infected, aren't the susceptible T cells all going to be wiped out eventually, and these would predominate?
But they gotta live though. They gotta be able to divide.
They have to divide. That's right. Absolutely. Okay. So
in fact, the cells they engineer here divide, but in culture you may not need CXCR4.
Yeah, I think whether they use stem cells or some other kind of cell hasn't been decided yet. What are they using in this trial? Do we know? Let's see.
They got I think they were using primary
T cells. Yeah. So several phase 1 adoptive transfer trials of CD4 positive T cells treated with zinc finger nucleases in HIV-infected individuals. So they're just putting in T cells to see if they're safe because it's a phase 1, right? But this phase 2, I think you would do the same thing, no? You can't change the protocol. So they're going to get T cells. And I think the hope is you get enough engraftment, but they're not stem cells, right? They're not going to divide forever.
Right.
I'm just not sure about this.
Yeah,
one.
so this is out of our range.
Yeah, I guess the issue would be if you, I don't know if you get HIV infection of stem cells further back.
Yeah, remember Kathy Collins told us about that, right?
That's right. Yeah,
you do.
Yeah, so you're still hosed because you're still going to be producing new T cells that can be infected.
Yeah, that's true. You really got to get this because remember she identified the receptor, one of these chemokine receptors,
right,
as functional in stem cells for infection. So yeah, you have to get those, otherwise you keep making new ones, right?
Yeah, I think maybe if you wanted to go the next step in clinical trials, you'd have to go back to something that could renew.
Yep. Anyway, they don't deal with that here. They're just.
This is a great start.
This is principle.
Yeah,
I mean, basically, they show that the gene is disrupted with this zinc finger nuclease. It does inhibit HIV replication, as you would expect with CXCR4 preferring viruses. The CCR5 binding viruses are not affected, as you would predict. One thing that's very interesting when you infect CD4 T cells, if you've treated them with a zinc finger nuclease, eventually the whole culture is resistant, becomes 100% gene deleted or disrupted for CXCR4. Right? So that initially you only have 30 or 40% disruption, but if you infect, you eventually get rid of all the cells that don't have a disruption and you select for the resistant ones. Right? Is that correct? So they show all that in vitro. Then they also do a mouse model where they engraft a severely immunodeficient mouse. They're called NSG mice, which are the most immunodeficient strains of mice described to date. They don't have T cells, they don't have B cells, and they don't have natural killer cells. So you can engraft them with a variety of different cells. And here they put in these T cells. So they take the human T cells with or without the zinc finger nuclease, put them in mice. They get some engraftment, which means that the cells multiply, and then they infect them with HIV. The results aren't great. They're kind of marginal.
Right.
Because they say this isn't... In fact, what happens is they probably get viruses arising that can use CCR5. Remember, they've put into these mice CXCR4 deficient cells. So the virus that they infect just changes and says, I'm going to use CCR5.
Mm-hmm.
So that just underscores that you have to knock out both to really be effective. But the mouse model isn't perfect, and they acknowledge the effect on virus replication is quite minimal. So they have to use a simian model, basically, right?
Yeah, they have a sort of a transient effect here where at 14 days post-infection, the mouse engrafted with the CCR4 knockout retains, and infected with the CCR4 tropic HIV has a higher T cell count,
right,
than the one that's got the CCR5 knockout. Okay, so transiently the T cells survive better in that mouse, but then after 32 days there's no statistical significance. So it's a shaky response in the mouse. But everything else makes sense, as you say. The virus shifts specificity, the cells are being engrafted, and all that kind of stuff. So it's clear that all of the things that you would expect are happening. It's not clear that they have it worked out appropriately to be a real therapy. But then they got this problem with the mice getting graft versus host disease.
Yeah, right.
So they're basically saying, look, there's no point in us pursue. This is proof of principle, and the model is just not good enough to be worth pursuing much further at this point. We need a better model, but it's working well enough so that we can justify going into a non-human primate model.
Right. Which will be next. And in fact, they make some T cells from macaques and knock them out, rhesus macaques, and they knock them out for both CCR5 and CXCR4, and they get disruptions and say, okay, we'll use this model next, which I guess will be the next paper before they go into humans with this.
Right.
So I've really revealed my ignorance in immunology in this because of this issue of what sort of cells you can engraft and expect to live for a period of time. But it's apparent that you can isolate T cells and make a graft with T cells, and that lasts for a while. So they must be amplifying. So that works okay. You don't necessarily have to. I gather, some listener can correct me, that you don't have to target stem cells to do this.
But I thought T cells would not limitlessly do that. I don't know.
We're going to have to go check on this.
Yeah,
we're going to have to go check on this.
If there's an immunologist in the audience, and I'm sure there is, they should set us straight on all this.
Yeah, well, the mice get engrafted with T cells, and they say one of the problems with the mouse model is, in fact, that they don't have normal functions. The human cells don't have normal trafficking and functioning in the mouse. So maybe that is something that happens in humans or non-human primates. But yes, so the question is, if you take T cells from a human, modify them, and put them back, will they multiply limitlessly?
Right.
Or were they? I seem to remember we had this conversation with Kathy Collins, because it's the same issue. She's doing very similar experiments. Anyway, this paper is wonderful. I think this is really cool. It's amazing technology. And one final thought is, we've been talking about a lot of cool papers in HIV lately, and it's clear that a huge effort is still going into trying to figure out how to deal with this.
Absolutely.
And more so than in any other virus infection, I would say.
Well, and that's because the target is already so well characterized and is so tantalizing in the sense that it seems like we ought to be able to do better.
Right. Well, of course,
and also, it's an enormous burden on global public health.
Well, and the drugs are only temporary, right?
Yeah.
They don't get rid of the virus. Okay, let's do some emails. This will be a long episode, but since we're going to skip one, you could split it into two. Listen.
There you go. That's what you do with your week off.
That's right.
To it twice. All right. So I had a letter from Mary. This was about live blogging. And I thought this would be good for Alan to address. She says, what's the deal with embargoes? Anyway, these are press embargoes. I know it's called the New England Journal rule, and I suspect it has to do with perhaps the press or big business somehow taking off with an idea before its time, but I haven't seen that ended by this practice. In my fields, history and economics, we freely discuss research and progress. In fact, that's when the most good work happens, when you have to answer questions or deal with stuff you hadn't yet thought of, or somebody has a new idea or new evidence or knows about a pertinent study you missed. It's very common to cite published manuscript or work in progress, or if I get a good idea and some leads from someone else's work and they haven't published yet, we would make up a name of a working paper I could cite in case someone wanted to know more about that person's work. And since I've been very sick for 2 decades now, a lot of my work circulates in that form with my permission. Why is medical science so different from other fields? I guess I've seen some of what can happen. A chance comment by Dr. Alter at a conference somewhere in Central Europe ends up in a story in a Dutch newspaper, and suddenly all hell's breaking loose in the U.S. But I guess I'm not sure that it should have led to all hell breaking loose. Maybe it's nothing more than that the press and the insurance industry and big pharma and policymakers all care about your work and really don't give two hoots about economic history, so you have to be more careful, but I still don't understand it.
This is kind of the same thing as it's related to the Engelfinger rule and stems from a similar concern. The idea was you want the papers to come out in a peer-reviewed journal and have that be the reference point for public discussion about the paper. And in an ideal world, that's the way things would occur. The idea of an embargo is that you take this paper that's about to come out in a peer-reviewed journal and maybe a week or so in advance, let reporters take a look at it so that they can go ahead and work up stories, and they have some lead time to work up stories that would hopefully be thoughtful and well-sourced, that could then drop as soon as the paper comes out. So that's the theory behind it. The difficulties are some of what Mary points to, and also things like embargo breaks, where either accidentally or deliberately somebody releases a story early before the paper is out. There's also I have had to sign a bunch of documents saying that I don't trade in securities, or if I do, I'll be careful not to violate the Securities and Exchange Act of 1930, whatever, which is insider trading. Because there's concern that if you have a certain population of people who have access to this information a week before it's going to come out, and in a lot of cases, this could be stuff that's price sensitive for small biotech companies, especially. How do you keep people from engaging in insider trading? And there are some measures in place to deal with that. But the reason for the embargo concept is to try to get good science journalism out there by giving reporters advance notice of the paper before it's generally available. That's the theory.
Yeah, it's interesting. Some fields don't publish in peer-reviewed journals. So I have a colleague at Columbia, Raul Rabadon. He's actually been on TWiV. And I was talking with him about this, and he used to be a physicist.
Right.
He says in physics, you don't use peer review. You just put your papers on a website, and people read them. And when you come up for tenure, the tenure committee downloads all your papers, and they read them, and they decide whether you've done a good body of work or not.
Right.
So I guess every field is just different. It's just evolved that way. But it's curious that I like that model where you just put your work up, and then when it comes time for the decision, they have to read your paper. And they don't just care about where it's been published, which is the way biology is now. If you got 10 cell papers, you might get tenure, whereas if you don't, you might not. It makes more sense, right?
Right.
Yeah. Yeah, but the problem with embargoes, or the reason for embargoes, is because a lot more people tend to be paying attention to medical research, which, again, Mary touches on that.
Yeah.
And this is the stuff that makes front-page headlines. If you look at the science coverage, or what passes for science coverage, most of it these days is actually medical.
Yeah, that's right.
It's what people care about. It's what people are going to read about. It's what they're going to talk about. And so these are going to be the big news stories. And how do you try to get those news stories to be well-researched and well-sourced and intelligent when the paper comes out and everybody expects that all the news will be about that at that time?
Right.
But as I said I think a couple of weeks ago on TWiV, if you just did away with press releases, you wouldn't have this problem, because it would take everybody a couple of weeks after the paper came out to figure out what the paper says, and then to do those stories.
That's a good idea.
And the general public is not reading the table of contents of PNAS and Cell and Science. So you could just have these papers come out on the regular peer-reviewed schedule. The reporters find out about them at the same time as everybody else, but the reporters would actually be hopefully paying attention, some of them, and would pick up some interesting papers and say, hey, I'm going to do a story on this. And then they'd have time to research and write it, because really nobody noticed when it came out.
Now, you would be among the first to come out with it because you understand it very well.
I do follow the. In fact, I look at the tables of contents of the embargo journals that I get, and I also look at some of the ones that I just check as they come out. And I often see stuff that, hey, it's kind of an interesting story, and nobody covers it.
And press releases come from basically people promoting their own stuff, right?
Yes.
So
yes, which is why you can't eradicate them.
Yeah, but it's also it's a strange selection for what's newsworthy.
Yes,
it's ego selecting for what's newsworthy.
It's ego, and it's also topic specific, and it depends on how the press release is written. I've seen stuff come out with poorly written press releases that nobody notices, even though it's I think would be perfectly newsworthy work. And sometimes you'll see that stuff show up in the news a month later. Mm-hmm. Someone says, hey, there's new research we've learned about. And I look at it and I say, you just learned about this?
Right.
I read the paper a month ago, and somebody just finally figured out that it was interesting because the press release was so poorly written. And unfortunately, a lot of what passes for journalism these days is really reading the press release and building a story from that.
I can't tell you how many times we'll do a story on TWiV, and then 3 months later, it'll hit some press somewhere, and they get it wrong. And I just think, if instead of reading just the press release, if they had looked through TWiV, oh, look, here's an episode entirely on this paper, and listened to it. I guess they don't have time to do that.
Exactly. There's an enormous amount and growing pressure on journalists, especially for daily outlets and instantaneous outlets online, and the TV and radio news and such. They need to produce stuff by 5 o'clock today.
Yeah.
And even if the paper came out a month ago, they'll pick it up and they've got some editor screaming at them saying, we need a story on this now.
Yeah. So we have to get rid of all the editors, basically.
Well, then you have other problems.
Yeah, that's right. Well, according to Dave Tuller, if the editors know science, that can be a real help.
Yes.
Because they can fix the mistakes.
Yeah.
Wow, very interesting. Well, I never had a press release about my work. Have you, Rich?
No.
No. So, I mean, I did...
Well, are you sure?
No, not really. But don't they usually ask us to look at it first, Alan?
Not necessarily.
No.
A lot of journals generate their own press releases. In fact, the very first thing that I did as a science journalist, I did an internship in the Nature offices in New York.
Yeah, I remember that.
And the first thing that they gave me was, here are the papers that are coming out in our next issue. Write a 2-paragraph press release about each one. And I was not expected to call up the researchers. I was expected to read the paper and then write a press release from that. And it's an intern's job.
Yeah.
So that's how those get generated in many cases.
Is there any way I could find out if there have been any press releases? Do they disappear?
They generally have a pretty short half-life.
Yeah. Okay, I'm not going to spend time. I just don't think there have ever been any, because I've never been asked to edit them in any way. And the work I did was a long time ago that might merit a press release, and I don't think they did it back in those days. So, here's an example. My first interesting result, the creation of an infectious clone of polio. David Baltimore called up his friend at the New York Times, Harold Schmeck. He said, Harold, we have a good story. And he says, let me hear about it. And he wrote an article. Yep. That's before press releases, right? The buddy system. Cool. All right. Let's move on to two questions from my virology course. The first one is from Eileen. It's fascinating that a virophage would so benefit algal populations in Antarctica, as discussed in TWiV 128. I wonder to what extent these virophages can be used in conjunction with current environmental science studies to mitigate global carbon dioxide levels. As you may know, carbon dioxide concentrations are fast approaching 400 parts per million in our global atmosphere, as measured in labs in Mauna Loa, Hawaii. One of the mitigation options environmentalists have proposed to use involves the cultivation of algae blooms in suitable bodies of water to not only fix carbon dioxide from the atmosphere, but also perhaps use the biomass as a form of fuel. Of course, the dilemma lies in algae proliferation in the absence of excess phosphate and nitrogen. What are the possibilities that a strain of this virophage can be created so that it can be used in temperate zones to help this carbon dioxide mitigation option? You got any thoughts on that, Alan?
I'm pretty pessimistic about all of these large-scale geoengineering projects.
Mm-hmm.
If you think about it, we're already engaged in a large-scale geoengineering project, and it's going horribly wrong. And that's why we have 400 parts per million of CO2, or soon will. So, and there's also been a lot of work on this stimulate an algae bloom, dump a bunch of iron filings in the ocean, and it'll fix global warming type of approach. And people have been working on that, and it turns out when you do these sorts of things out there in the real world, they don't have the kinds of effects that you'd expect. Because, surprise, surprise, the ecology of the Earth is more complicated than we understand. So in terms of coming up with a virophage that would change the algae balance in the ocean and maybe do that to a degree that could bring CO2 levels down in the atmosphere, I'm very pessimistic about that. I really would not expect an approach like that to work, because once you take your engineered virophage out and you throw it in the ocean, it's going to be competing with all the other virophages out there, or all of the other organisms out there that are either going to eat it or compete with it, or it's going to get integrated into their genomes and mutated, and it's going to return to the balance that we had.
Right.
Yeah, the virophages out there have had a lot longer to evolve than the one you created.
Right.
Yeah, we talk about this all the time. Nature's already done the job. It's not nice to fool Mother Nature, right?
And if there were some adaptation that would, there will be adaptations to a higher CO2 level, and we'll see them, and they'll arise naturally. Mm-hmm. And they will completely dwarf anything that we're able to do. Whether they'll actually back off the CO2 level far enough to make a difference to this whole infrastructure that we've built, based on the current sea levels and all that, I don't know. But those experiments are already being done, and not by us.
Whatever those forces are, don't necessarily care about the future of humans on the planet.
Exactly.
Okay? It's just going wherever it's going.
Yeah, and it's worth pointing out that if you look at the geological history of the Earth, sea levels and ice extent and all these things have varied over vast distances at different times. And, sometimes people will say, oh, so why are we worried about global warming? And the answer is, if we had glaciers extend down to North Florida all of a sudden over the next 100 years, then all this infrastructure that we've built north of there is going to be really in deep trouble. So there's an adaptation that we would have to do that would be very disruptive.
As long as it doesn't get to Gainesville, that's okay.
That's right.
And I wouldn't mind moving a little south anyway.
It can't go much further, can you?
Oh, I can go down to Miami, I suppose. I'd like to go to the Keys.
Yeah, that's good. I hear that's
nice.
Nice.
But if the sea level rises, you definitely don't want to be in the Keys.
Dang.
I was down in Key West, and there was a thunderstorm, and it practically submerged the place.
Next letter is from Anne Marie. My question to you is related to the many posts you have had about poor coverage of scientific issues in journalism. Although David Tuller is beginning a new dual program at UC Berkeley, do you think undergraduate courses could also give the needed scientific background instead of a master's in public health? Clearly, every journalist writing about health-related topics will not have a major in biology, but do you think that perhaps requiring science classes in a school's core as an undergraduate could also help minimize this problem? And if so, what specific science classes would be most useful, either as an undergrad or while pursuing an MPH? I suppose my overall question is, do you think that this dual program is really the best option, or is there a better way for journalists to acquire a scientific background? We actually saved this for you, Alan.
Got a whole bunch of letters from me here.
Yeah.
Do you want to read Dave's reply first?
So Dave Tuller answered. He was on TWiV a while ago. So he's a journalist out in San Francisco. That's interesting. Thanks for sharing. My answer would be any educational work in these areas would be helpful. Adding on the MPH is just one way to do it. Certainly, if a student was a public health major as an undergraduate or had a lot of biology, microbiology coursework, that would be a good basis for covering public health, infectious diseases, etc. as a journalist. The MPH, I think, does add a professional component. You start to think the way someone working in public health would, and that's a very helpful ability to have as a journalist.
Also, some journalism schools, and I'll just give a plug for one that I used to teach in at NYU, but there are others. NYU has a program called the Science. It's now the Science, Health, and Environmental Reporting Program, SHERP, and it's a master's program in the journalism school. They get roughly a dozen students a year, and it is designed to turn out science and health reporters, science, health, and environmental reporters, and it does so with a series of courses aimed at people who are either coming from kind of a journalism background and want to bone up on the science, or more frequently, they're getting people who majored in a science and want to go into journalism. So when I was teaching it, the majority of the class was typically folks who had at least undergraduate degrees, and usually there'd be one or two PhDs in the course as well, who had very, very solid science backgrounds, but they didn't know how to write a story, putting in a lead and structuring the narrative of a story, and also the SHERP program teaches things now like online reporting and multimedia reporting and how to use video and all this sort of thing. So there are programs like that will either provide that provide the missing component. They take people who need the science and they teach them a bit of science, and they take people who need the journalism aspect and already have the science, and they teach them the journalism. And those are a great way, I think, to turn out reporters who can do this sort of thing.
And of course, all future journalists should take my virology course.
Yes, of course.
And you can, because it's online.
That's right.
But of course, it doesn't cover all health science, and that's a problem. But
no, but I think going into some depth in at least one field is very, very useful across fields. Because what would be more useful than a particular body of knowledge, somebody knowing a particular aspect of public health or knowing virology or knowing bacteriology, what I think is a lot more useful for a journalist is understanding how science works.
Yeah, that's a good point.
If you have that, then you know how to approach a science story, and you can go from there.
Hey, they should take my course and listen to TWiV, and they'll be pretty well
Absolutely.
off.
Absolutely.
Okay. The next one's from Neva. She sent a link to a site which is using search patterns to track dengue fever. So this is like Google Flu, I guess, where they use search patterns to predict influenza outbreaks. Here they're looking to track dengue fever. And she says, more tracking initiatives. I notice they do not mention tracking dengue in Central America, though. Now, why would that be?
It doesn't make any sense to me, because the way this works, okay, is that Google just surveys the use of Google.
Yeah.
Looks at search trends, okay?
Mm-hmm.
So they use a bunch of different, keywords, I suppose, and look for the distribution worldwide of people that are looking for are searching for things related to dengue. And what they've shown is they actually have data here. They got graphs that show that the frequency of searches for dengue stuff correlates with peaks of dengue activity in different places. So I don't know that Central America is specifically excluded from this. I mean, it would get anywhere.
Right, and they seem to have rolled it out in only a few specific countries. They have Bolivia, Brazil, India, Indonesia, and Singapore.
Yeah, they have available data and adequate search volume in those countries.
Right, and I think the issue is how much penetration do the internet and Google have?
Actually, that's interesting. Yeah, that's interesting because there is a bias in this, because it assumes, in order to be completely accurate, it assumes that everybody everywhere has the same sort of frequency of use of Google, and that's not necessarily true.
Right, Google is not the only search engine. And not everybody has internet access,
Right.
and in a lot of countries there are other search engines that are more commonly used.
Right. Okay.
So that may be a big part of it.
Nevertheless, it's interesting.
It's cool. And there's an article in PLOS Neglected Tropical Diseases about this, which we will link to. Thanks, Neva. Should we keep going, guys?
What's our time?
It's 3:43.
Oh dear, I actually have to get going.
Okay. Do you want to do your pick quickly?
Yeah, let's go ahead and do that.
Sure.
So my pick this week is a site called Bugscope, actually a program called Bugscope, and this is at the University of Illinois in Urbana-Champaign. And they essentially give an electron microscope to school children.
Wow.
Now, you don't actually get it shipped to your school and set up, but what you do is you take your class, and this could be elementary school all the way up through college, or as they say, K through 16.
Okay.
Now, presumably at the college level, a lot of folks would have access to facilities that they're on their own campuses, but not necessarily. But it's really aimed at K through 12, I think, mainly. So you send the class out, you say, collect some bugs. And of course, insects are everywhere. So the kids go out, they get some flies and some ants and what have you. You stuff them in a tube and you send them to these folks in Illinois. Now you have to apply for this program first. You don't just randomly send them bugs. But what they'll do is they'll have a time slot, and it'll be timed, hopefully, to coincide with your class. So your class meets, and there you have your view in your web browser. And they have some sessions that they've archived, and you can see what's going on. You have a chat session, and you have controls for the microscope. And they'll put one of your bugs in the electron microscope, and you can control the microscope and look at your bug.
Neat.
These are scanning electron micrographs. The nice thing about insects is they do very well on EM, and there are lots and lots of tiny little details to look at. So it's a real. The kids are driving the scope within certain limits, and they can scan around and look at the parts they're interested in, and these are the bugs that they caught.
Scanning electron microscope is really cool because the preparation is so easy. You just stick this thing on a stage and plunk it in the scope, and you get to look at the surface features, and it's just really dramatic.
Yeah,
really cool.
And they have some of these. They have these images archived here on the site.
Yeah,
very neat. Cool. That's great. All right, Alan.
Yeah.
All right,
Thank you very much.
Have to get going.
AlanDove.com. Thank you for joining us.
See you, Alan.
Always a pleasure.
Bye-bye.
All right, take care.
That leaves you and me, Rich.
I guess so.
Should we continue with our picks?
Sure.
What if there's that wonderful Skype goodbye noise.
Huh.
What is your pick?
Okay, I have picked a commentary from 1998 called Vaccine Adverse Effects, Causal or Coincidental by Robert T. Chen and Frank DeStefano. What is special about this? It is a commentary that appeared in Lancet in exactly the same issue as the famous Wakefield et al. Article that concluded that there was a link between the MMR vaccine and autism. And this commentary, completely published in the same issue, completely debunks that article. Basically goes through a bunch of different reasons why that article is quite likely to be not true. And it's very well reasoned. These guys are vaccine safety experts and epidemiologists at the CDC who wrote this article. And they wind up concluding, this is great. Effective and credible systems are needed for the detection of vaccine-associated adverse events through pharmacovigilance, for distinguishing causal reactions from coincidental reactions by pharmacoepidemiological or other studies, and for risk communication. Without such a system, vaccine safety concerns such as that reported by Wakefield and colleagues may snowball into societal tragedies when the media and the public confuse association with causality and shun immunization. This painful history was shared by the UK, among others, over pertussis in the 70s, after another similar case series was widely publicized, and it's likely to be repeated all too easily over MMR. This would be tragic because passion would then conquer reason and the facts, again in the UK. And that's exactly what happened. There was panic with the Wakefield article, and vaccine coverage dropped in England, and people have died as a result. And what's remarkable about this is it was in the same issue, and it was apparently ignored.
Yep.
At any rate, this ordinarily is behind a paywall, but I've had some communication with. I wanted people to be able to read this, so I've had communication with. Actually, it turned out to be ultimately a deputy editor at Lancet, who has kindly agreed to make both this and the Wakefield article publicly available. So there'll be a link in the show notes, and if you click on that link, it'll take you to the article, and you have to register with the Lancet, but it's free. And there's no penalty or anything else, and you can get the article, and you can read both the original Wakefield article, or if you're interested in this commentary. Ordinarily a commentary is something that maybe your experience is different, Vincent, but usually it's written by somebody who was either recruited by the editors or maybe was a reviewer of the work or something, and usually a commentary is written to hype the thing.
That's
right?
That's right.
And this is a commentary that absolutely bashes this. It's puzzling. I almost don't want to know the backstory on this.
Makes you wonder why they would publish an article if they had to include a commentary such as that.
Well, I have seen situations where a commentary is written. Matter of fact, I've been involved in one of these, where a commentary is written that does insert at least a small note of caution here, when there was a lot of controversy about the reviews of an article. Okay, so that it's like this might be true, and it would be a shame not to publish it if it is, but we want to cover ourselves in case it turns out to be not true. Right, so we're going to publish this this commentary that's a way out. Now, I don't know what happened in this case, but maybe something like that. At any rate.
Well, of course, we know the article's been retracted by the journal and by all but one of the authors. Right.
And basically there's accusations, at least, of fraud on behalf of the investigators. But it took them, 20 years to figure it out?
Yeah, it's very sad. And now we have measles outbreaks everywhere as a consequence.
Right. And if you actually do a PubMed search for Chen and DiStefano, you see that there's a whole series of publications where they follow the whole MMR thing over the period of years and publish on the subsequent epidemiological studies that were done that further debunk the original link. But at any rate, I thought this was historically very interesting, and I wanted people to be able to read it. So
yes, and you should be thanked for your persistence at getting this opened up.
Well, it was an interesting journey, but in the end, when I got in touch with some humans, okay, they were very reasonable humans and did the right thing, so that's good.
So Alan has written about this at his blog, alandove.com, I believe. He wrote about your initial rejection.
Right.
Right, and then, so we'll put the link to that as well, so people can see the automated reply of no, and then finding a human and getting them to listen.
Yeah, I do want to communicate the bottom line that once the humans were contacted, there was no problem. Okay, they were very, Very understanding.
Yes,
Okay?
yes, humans can be helpful.
Right?
More often than not. My pick is a book called West Nile Story by Dixon de Pommier. Now, you may ask, why am I picking this? Because it's an old book, and it's by Dixon, who wouldn't want us to pick it, but he's not here today. If you haven't read it, it's a very interesting recount of the original West Nile outbreak in New York City. And Dixon originally published this as a self-published book. He has a publishing company called Apple Trees Productions, LLC. He printed 12,000 copies of this book, and he sold them all, and you haven't been able to get it for a while. You can get used or third-party resells, but you haven't been able to really buy it. And he said to me about, 3 months ago, can we make an ebook out of this? So I said, sure. He gave me the text, and I formatted it and put it up on Kindle at Amazon.com. And I think it's $3.99. It's pretty cheap.
Cool.
So check it out. Okay. I think it's a cool story written in his style and worth reading. And at that price.
So tell me again, how you facilitated this somehow?
Yeah, because Dixon's wouldn't do it himself. He's not very tech savvy. He said, can we put my book as an ebook? So I said, sure. I looked up online how to do it. He had we made him a Kindle account, an author's account on Amazon. And I found instructions. It's not hard.
Wow.
You have a Word document. You can just format it in a certain way, and you upload it. They convert it to the EPUB Kindle format, and it works just fine.
Great.
And,
so has it got illustrations in the whole bit?
So this does not. The original one had just a few drawings at the beginning of each chapter. Now, the next book he wants to do this with is his parasitology book.
Right, that's got all kinds of illustrations.
Yeah, and they're in color, so it's going to be harder. And I don't have time to do that right now. But eventually we'll do that as well, because that's also self-published, and he's out of those. So we're going to do that. So this was an experiment, and it seems to have worked. So next we'll work on the images. As you know, the Kindle is black and white, but it can be read. The Kindle format can be read on other devices, I believe, and such as an iPad.
I can read it on my iPhone.
In which case, if you have color in it, the images show up in color there. So if you read it on a Kindle, they'll be black and white and look crummy, but on other devices, they'll look pretty good. So when I get some time, I will do that for him. But in the meantime, check that out. Make Dixon happy. Buy a copy. And that will do it. We don't have a listener pick of the week. You guys have to send some in to us, okay? TWiV at TWiV.tv, along with all your other questions and comments, of course. You can find TWiV at iTunes at the Zune Marketplace. We have an app that you can use. I think you use that app, right, Rich?
Yep.
To stream the episodes. It's MicroWorld app.
Yep, I have done that.
Microworld.org/app. You can get it for Androids or iPhones. Or you can go to TWiV.tv and listen and look at our show notes as well. And as I said, send us your questions to TWiV at TWiV.tv. Rich Condit at the University of Florida, Gainesville. Thanks for joining us.
Sure enough, and I'm going to over our next recording is 2 weeks from now. Between now and then, I'm going to go talk to some of my immunologist friends and straighten this mess out and correct it in the next episode.
I actually got an offer from an immunologist to join us now and then to help us out.
That would be good. That would be good.
So
I didn't really anticipate that we were going to run up against this in this one. I didn't really think it through carefully enough, but I'm sure that there's a reasonably clean answer to this. But of course, immunologists are kind of like economists. Any question you ask them, the first thing I say is, well, it depends.
Yeah, it depends. Whereas with nucleic acid, you ask what the sequence is. And
ACTG, you know?
This is it.
Right.
So, yeah, immunology is a squishy science.
Yeah, but it's great.
It is wonderful,
The more I learn about it, the more fascinating it is. It's really great stuff.
I like immunology. There you go. I'm Vincent Racaniello, and I'm at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back soon. Another TWiV is viral.